Mashad Warjai. Hello and good morning to everyone. I'm Minu Singh, faculty from Kathmandu Research Institute of Biological Science. Today we have presenter Mr. Bijay Raj Khanal with us in Biomedicum Seminar Series Episode 5, which is jointly co-organized by Kathmandu Research Institute for Biological Science (CRIPS) and Research Institute for Bioscience and Biotechnology (BIP). Before moving to introduction, I would like to go with few housekeeping rules. The presentation will be nearly of 30 minutes, and then the session will be open for question, answer, and discussion. The session is being live streamed on social medias and is being recorded for educational and training purpose. Not further delay, I would like to introduce our speaker for today. Our speaker is Mr. Bijay Raj Khanal. He is a senior scientist and digital pioneer at AstraZeneca. He has completed his BTech in biotechnology from Kathmandu University and Masters of Biotechnology from California State University. And also he has completed a certificate course in system analysis from University of California in Los Angeles. He has gained a long experience of more than 8 years in development of biologics, focusing on cell line development and cell line engineering to deliver high-titer, high-quality serum-free antibodies in various formats like monoclonal antibody, fusion proteins, and others. Likewise, he is also expertise in clonal screening, scripting, cell line development production method, advanced microscale in bioreactors, and other relevant fields. So, without further ado, I would like to hand over the session to Mr. Bijay Raj Khanal for his presentation. Thank you.
Thank you so much for the introduction. Hello, everyone. My name is Bijay again, Bijay Khanal, and today I'm very glad to talk about overview of cell line development and manufacture. And then I would like to use this opportunity to thank CRIBS, the whole Biomedicum congregation, for providing me this opportunity to share some of my knowledge and my skills so that a lot of people, I'm hoping, can benefit from it. Okay, so in this talk today, my overview, my outline is as follows. I will start with biologics introduction at the beginning. We will go through a little bit of overview of your drug development process. And then we will get into cell line development in particular, which is my expertise and something that I have been doing for quite some time. And then we move on to manufacturing, where I will give you a short overview of how the manufacturing of drug development looks like. And then I will also end my talk with some of the automation and some process analytics in cell culture. And then I will be up for any questions you have at that point. All right, so moving on. So my aim, the aim of this talk is, like I said, a basic overview of mAb development process focused on cell culture and cell line development, which is the part, which is my expertise and the part of my work that I have been doing at AstraZeneca for over 6 years now. And then with this talk, I would like to disseminate information about various techniques used within the industry, what the skill set is required for people who would like to join this kind of industry or who would like to gain some sort of like knowledge about how to proceed in drug development, and then also provide some scope and opportunities for all the listeners out here today. Okay, so since my introduction has already been done, I'll skip this part. It's only like whatever it's on my resume. So I will just move on to the nitty-gritty of things. So I would like to begin with a high-level overview of drugs. So what exactly are drugs? So drugs, also known as medicines, right? Also known as therapeutic agents, anything that fixes the harm done to our body by diseases, whether it's infectious diseases or like any other kind of um, life-threatening diseases such as cancer. Drugs are the ones that people take to rectify the, like the immune system and recover from the damage, right? So broadly, drugs can be divided into either small molecules or large molecules. So the small molecules are the ones that you can find in normal pharmacies, like tablets, for example, aspirins or paracetamol, you name it, right? All those different small size tablets or syrups, they are all small molecules, right? And by the name itself, the small molecule drugs are smaller in size. They are less than 1 kilodalton in most cases. And then there are large molecules, also known as biologics, right? They are larger in size, usually less than or equal to 16,000 kilodalton. And some of the examples are your cancer drugs, such as Herceptin for breast cancer that Genentech makes, Humira that AbbVie makes for some autoimmune disease, right? So the biologics may be of different formats. It could be monoclonal antibodies, bispecifics, duet maps, which are type of biospecifics, fusion proteins, T-cell engaging domains, or like the very popular ones, CAR-T, which are gaining a lot of momentum these days in the field of biologics, right? So here I have a small, you know, a schematic to say about what different kind of biologics are there. So you have your MABs on top here, which are targeting the cancer cells or tumor microenvironment using various techniques like disrupting the immune checkpoints or doing different mechanisms like ADCC or CDC. ADCC is antibody dependent cell cytotoxicity, or CDC is complement dependent cell cytotoxicity, right? So various mechanisms are used for the MABs. And then you have your antibody drug conjugates also, where you have your MAB, then it's conjugated with a drug, which is also called payload, basically. And then when the antibody binds to your target cell, then it releases these payloads and causes the disruption and killing of the cells, right? And then you have your bispecific antibodies, where the two Fab of the antibody, as you know, this on the Y-shaped antibody, the tops of the Y are the Fab regions, and the bottom is the constant region, which is the FC region, right? So the Fabs, each of the Fabs can connect to attach to a target cell as well as some other components such as T cells, and then disrupt the cells. And then you have your CAR T cells, which, like I said, is a very new technology where the blood of the patient is taken out using a process called leukapheresis, and then it's excited. The T cells are isolated. The T cells are excited with your with your with your product, your molecule or your therapeutic agent, and then it's injected back into the patient to cure diseases such as leukemia. And I believe only a handful of CAR-T therapies are available in the market right now, and it's very, very expensive. But this field keeps on growing as we speak. Okay. So if there are any questions at any point, feel free to interrupt me. Okay. There's no, I will not feel offended or like I will not be disturbed by it. Okay. So see, the drug development And from now on, when I say drug development, I will only talk about biologics. So AstraZeneca makes both small molecules and biologics, but my field and most of our field, I believe, is in biologics. So I will only focus on that for the scope of this talk. So yeah, so basically this is biologics development. So biologics development is a lengthy and expensive process. It takes like 10 to 15 years of work in R&D, manufacture, toxicology studies or tox studies, and clinical trials. And then you have to do your all your different regulatory filings, etc. And then it takes a long time to do all that. And it's also very, very costly. So each molecule that goes from research into the trials, clinical trials, and into filing costs like around $2 to $3 billion. And the approval rate as of 2000s was really, really low, only 5% approval rate by the FDA, because most of these drugs fail in clinical trials. But that has significantly improved now because of the improvement of the way in which our therapies are, like measured for safety, efficacy, and also our statistical, you know, design for clinical trials has also improved a lot, and that's why it's growing now. But it's a very expensive process. So that's the bottom line for now. All right, so different phases of drug development. So you have so in this previous one, as you saw, there's research and early development, then there is development, then it moves on to manufacturing and trials, right? So when I talk about research and early development, there's various different things happen within early research. So that's target evaluation, target validation, pre-project, lead isolation and optimization, and lead candidate profiling. These are all research steps where initially the target is studied, right? So what is the unmet medical need that we are trying to fulfill, right? That's studied. What is the method of intervention that we are going to put in, right? Whether we can, apply these scientific rationale that we have about these intervention that the target can affect the disease, right? So all that needs to be studied and validated as well. Mechanism of action using various bioassay models is also need to be done at this stage. Also potential and feasibility, all those kind of things need to be evaluated at that point. And then you have your pre-project where you have a candidate drug nomination. You come up with all these different formats of the drug. Then you have your main molecule that you want to nominate and then move forward with one, right? Because you cannot side-by-side progress all of that because it takes a lot of time, money, and then resources to drive that through the pipeline. So that's why you want to nominate your candidate drug in the initial stages. Then you want to stick to a molecular format. You want to study about the specificity of that. You want to study about cross-reactivity, and then you want to study about pharmacokinetics and pharmacodynamics, basically to ensure that it has a good shelf life. It's not a drug that needs to be given to the patient every week or so because it's an IV injection or IM injection. It's very difficult for the patient, so it should have a considerably long, dosing regimen, and then it should be fairly safe. At the end, it should be safe. So that's the main idea of the drug. It should be safe. It should be efficacious, right? And then you have your lead isolation and optimization phase, where you have you identify the key properties, right? What's in your drug, right? You want to identify how stable it is. Is it suitable? Is there like any safety risks that you can mitigate in advance by tweaking some of your DNA sequences or doing some sort of adding something or deleting something that can cause less safety risk, right? And then expressibility. So for the drug to be developable and profitable, it should be expressible, right? It should have a decent amount of antibody concentration when it enters the cells, and then you should be sure enough that that can be profitable to the company, because at the end of the day, every company wants to make money out of these expensive drugs, right? And then you have lead candidate profiling, where you deliver the candidate drug to other group, and you have your safety and efficacy studies, and then you ensure that your manufacturability criteria is met. Okay. So moving on to development and manufacturing. So this is more like my part of the job that I do here. So I'm mostly involved with preclinical development and going into phase 1. So in the preclinical development stage, you have your drug candidate identified and approved, right? And then you perform animal studies, small scale tox studies, make sure your mouse doesn't die after giving 1 dose of your drug, right? Make sure it's stable, and they show some sort of, like efficacy of the drug, right? And then you have all your, different steps that are involved with regulatory, with making sure, like the CMC is chemistry, manufacturing, and controls. So that's a big group, a development group, which has, like leads from all different areas: cell culture, purification, analytical, formulation, regulatory, everyone, and they constantly discuss about like how the molecule is moving through the pipeline, what's happening, is there any risk, what to do about the risk, how to fix it, all those kind of things, right? And then you have to prepare for the initial regulatory submission, which is the IND in the US and CTA in the Europe. So IND is investigational new drug. That's for the US side. So once you are ready for phase 1, you file the IND, and then you proceed with all your clinical trials and such. And then you also have to develop something called an FTIH protocol, which is first time in humans. After animal studies are done in preclinical development stage, all the other steps, phase 1, phase 2, phase 3, will need actual human patients. So you want to make sure like your protocol is developed, everything is in place, everything is covered for. Okay. In phase 1, phase 2, and phase 3, and this is one of the longest phases in the development and manufacture of antibodies. You basically do all your development work, right? There'll be cell culture, there'll be purification, there'll be analytical methods, there'll be formulation development, there'll be regulatory, manufacturing sciences are also involved, and those kind of things. As the process development is happening, the clinical development is in parallel, right? Where in phase 1, you have about 10, 20 patients, and then the only reason to do phase 1 is stability, scalability, just making sure that that's covered, right? And then if it passes that phase, you go on to phase 2, where you have to enroll hundreds of patients, right? Where you study robustness, how like robust is the molecule, how robust are your cells, which develop your molecule. All those things are studied at that point. In phase 3, you have thousands of patients. I've seen 2,000 to 20,000 patients enrolled in phase 3. So the idea of phase 3 is for safety, efficacy, and that should be with high statistical confidence, right? So your phase 1 and phase 2 are more like your, how to put it? It's more like you can still come back and forth, and you can still evolve your cells or evolve your methods during these phase. But phase 3 is like at phase 3, you have to have a process lock in place. You cannot change your cells, or you cannot change your methods. Those cannot be changed, because at that point, your safety and efficacy is really, really important, right? And then you eventually submit something called a BLA, which is for the US. It's called Biologics Licensing Application, which is a like 3,000 pages, 300,000 pages long document that is submitted to the regulatory body here, which is FDA, the Food and Drug Administration, and then you wait for the response from them, whether your drug is approved or not, right? And then you have your commercialization phase, where you do all the last, like packaging, selling, those kind of works. But you need to ensure that your life cycle of the drug is maintained, and you have to maintain the life cycle of the raw materials as well. So for example, if a vendor says they used to make yeast extract for a cell culture, but now they don't make it anymore, then you need to go back and ensure that whatever replacement vendors that you find who is making those raw materials now, you have to bring it to the lab. You need to test it and make sure it's stable, it's robust, it's safe, and it's efficacious. So you have to maintain those that life cycle. And then there is continued process verification, validation, which is continuously looking at the process performance and making sure nothing is out of the specs that you put in place, and ensure like everything is in place, right? So that's a lot of theory, right? So let's get into some of the actual development. So for development, so looks like I have a question on the chat. Safety in phase 3 is not that in initial phase 1. Yes, safety is important in phase 1 and phase 2 as well, but it's also important in phase 3. I mean, like it's important in phase 1 and phase 2, but it's not taken as seriously in phase 1 and phase 2. Of course, safety is always important, right? Even from animal tox study stage, the safety is important, but safety is the primary reason when you are in phase 3. Okay. All right. So like I said, the part of the work that I do here is called cell line development. So why cell line development? What is the need for doing cell line development? So the therapies we deliver are monoclonal antibodies, right? So what are monoclonal antibodies? Monoclonal is 2 words, mono and clonal, right? Mono is single, clonal is isoform, right? So these are specific antibodies which are developed in clonal cell lines, right? And they are very specific to a target. So HER2, if there's a HER2 target, like your breast cancer will have a HER2, will express HER2, right, on its surface, or as an epitope, right? So your therapy that you make, which is anti-HER2, needs to go on and bind to that specific. It will only bind to HER2. It will not bind to anything else, right? So monoclonal antibodies are specific, as well as they are developed in clonal cell lines, right? So that's why it's very important to have cell line development, because the regulatory agency requirement, FDA, requires that every therapy, every antibody needs to come from a cell line that came from a single clone, meaning one cell, right? A single cell. It has to come from a single cell, pretty much. So why clonal cell lines? Why do we need to have clonal cell lines? That's because these are isoforms that have identical profile. They have predictable product quality. Again, for safety, efficacy, potency, and consistency. we need clonal cell lines, because whatever antibodies are given rise from one cell versus your multiple cell, they might differ in product quality. So that's why you have to have one cell line, right? So we've talked about cells. So what are the cells that are used in the industry? So mostly the cells that are used in the industry are CHO cells. 75% of all the monoclonal antibodies developed right now are done in CHO cells. Then you also have CHO is Chinese hamster ovary cells. Then you have HEK293 also. A lot of your AAV or adeno-associated virus, which are also growing these days, are made in HEK293, which is human embryonic kidney cells. And then you also have some other cell lines like NS0 cell lines. We also, some of the therapies are also made in bacterial cells, such as E. coli. For yeast, it's mostly Saccharomyces or Pichia, right? But why use CHO cells, right? CHO cells is suspension culture adaptable. It can grow in chemically defined serum-free media. It has a short doubling time of about 24 hours. It has demonstrated safety and quality, and it can replicate PTMs, meaning post-translational modifications. So whatever post-translational modification that you see in one round of culture, you can expect to see that in the next round also. Okay. So the process that occurs in cell line development is mainly going from receiving the heavy chain and light chain sequences from research. So cell line development is the first step in development. So there's research, and then there's development, and then there's manufacture, right? So after research, the first step is development. So the research hands over the heavy chain and light chain sequences, and then those heavy chain and light chain sequences are ligated into a backbone vector along with GS cassette to create an expression vector. The reason why you use GS cassette is you have to use some sort of selection in the media, right? Just like you use ampicillin resistant vector when you're growing your bacterial culture, right? You have to have some sort of selection for your mammalian culture also. So GS is the cassette that goes into the vector, and then your culture will have MSX or methionine sulfoxamine as the selection agent. Okay, and then you do a stable transfection into CHO host cell, in which the sorry about the blurred picture. Your DNA will go into the nucleus and integrate into the chromosome of the cell, and then start expressing your antibody of interest, right? It's going to export it out into the media, right? And then once you do a transfection, you have a population called pools, right? And I'll come to that in a little bit. We have to sort those pools to obtain clonal cell lines, meaning you have to have like your 384 well plates where you deposit one cell in each well of the 384 well plate so that you can start growing your cell line, your initial point of cell line from there, okay? And then you want to expand clones based on cell and antibody, quantity and quality, right? So basically, as you scale up, so at this stage, you have about 75 microliter of cell culture. So meaning like one cell is deposited into 75 microliter of media. But then you need to expand it, right? You need to scale it up as the process moves forward. So you want to go up to about 2 liters at the end of cell line development. So you go from 75 microliter onto like different plate, and then you move on to shake flask, and then into the bioreactors and so forth, right? After you sufficiently expand your clones in shake flasks, or other flasks, then you perform a clonal screening where you have all your clones in bioreactor, and then you do a clonal screen that mimics the actual production run. And then from that, you measure your cell culture performance, you measure your antibody concentration, and then you can identify the top cell lines to move forward with. Okay, and at the same time, you need to do various product quality and analysis, such as glycosylation, fucosylation, all those different kinds of things, all those post-translational modifications, as well as validation that your cell lines are actually correct cell lines. You have to perform your cDNA sequencing and those kind of things. And at the end, once you are happy with your top 5 to 6 clones, then you grow it up into 2-liter cultures or even more, and then you basically bank it into cryovials and put them in the liquid nitrogen. So at this point, in the beginning, you start with maybe 6,000 to 8,000 clones, right? And then you go all the way to top 6. So the whole throughout the way, you have your screening, clonal expansion, as well as screening through different processes, and you get to the end. So this whole process takes about 12 to 16 weeks of time. Okay. So again, to come to it, you can track where in cell line development we are on top here. So I'm going to go with the first one, vector engineering, start with the vector engineering. So initially, when they do the discovery, target discovery and protein engineering, they use different ways to identify your, target of interest, your antibodies, or the targets that you are going to be working with. It's going to be done using different methods like mouse hybridoma, which is very popular to develop monoclonal antibodies, right? And then you have phage display, you have PBMC, which is basically your plasma, isolating blood from your plasma, then RNA, and then do a complementary DNA sequencing, come up with the DNA sequences, right? So think of it as sort of like fishing. So you would have a panel of antibodies here, right? And then you would go fishing into the target, and the ones that bind to these targets, those are your potential antibodies of interest. And then you work with them. And then you move on to isolate and optimize them. Make sense, right? And then you do all the things that I talked about earlier, expressibility, right? You do transient transfection. You take a look at the antibody concentration from that. Transient transfection is fast. You know the results in 2 days. And, you can, choose and throw away the clones or the antibodies that you do not want at that time, right? Then you also stick to a format, you learn about shelf life, all those things. So all those things are optimized at this stage, right? And those sequences are then once they come up with the optimal lead sequence, they relay those lead sequences over to us. And these lead sequences are heavy chain and light chain sequences. And then the first step will be vector engineering, right? So once you receive these heavy chain light chain sequences, you want to do a restriction digestion and isolation of your fragment of interest, right? And then you ligate it, right? Typically, the ligation happens in this way. You have your heavy chain variable, light chain variable, heavy chain constant, light chain constant, plus other incidental components, including your GS cassette and your, origin of replication, all those different things, and then your polyase signals, all those things, and then you do those kind of ligation, and then you do a transformation, bacterial transformation, and then you prep your DNA, make as much pure DNA as possible. You use like endotoxin-free DNA prep kits to come up with your high quality, high amount DNA, and then you validate that DNA. Make sure the sequencing is correct, like do a DNA sequencing, make sure the DNA looks correct, run it on the gel, make sure it's the band that you're looking for, right? And then you purify the DNA with the phenol chloroform technique, right? Then you do linearization of DNA. So why do we need to do a linearization of DNA? Because it's a stable transfection. It needs to go into the cells. It needs to integrate to the chromosome. So that's why we need to linearize the cells. The major difference between having a DNA in a circular format, where you can use it as a transient, where you can use it for transient transfection, but for stable transfection, you need to linearize your DNA. Okay, and then it's over to cell line generation, right? So just want to show this schematic here. Everyone has seen this before. So like I said, the bottom part is your FC part, the top portion is your Fab portion, and then you have your variable regions here. So basically, make your Can you guys still hear me?
Yes, we can. And sorry to interrupt you, Mr. Like your time,
Yeah.
you're going out of time. Could you please try to wrap up the presentation?
Oh my God. Yeah.
I'm sorry for that.
Okay, no problem. Okay, I'll try to wrap it up quickly. Right. So then you have, so like I said, I'm going to go very quickly now. So you have your stable transfections where you transfect your cells, and then you come up with something called pools, which are your parental lineages. And then after a week or two, you look at your you do an intracellular staining on your cells and identify, so where you stain your cells with heavy chain markers and light chain markers, and then your untransfected CHO cell will be on your fourth quadrant here, Q4 here, but if it's double positive, it's double positive for heavy chain and light chain, it should be on quarter 2 here, just like this one. And this is a like not a very great one, but so that in that case we'll drop this pool and we'll carry this pool forward. Okay, and then we move on to generating the clones and clonal expansion, where you have your cell sorter, which sorts these cells into these 384 well plates, just like I said. And then you have your series of expansion steps where you go from 384 well plate to a 96 well plate, and then you go into suspension culture where you have your 96 depot plates, passages, and then you perform a fed batch run where you look at titer, you look at your cell growth, all those things, and then you scale it up continuously, and then go on to these bioreactors, and then perform product quality analysis, and then eventually choose top 6 based on specific productivity, titer, antibody concentration, robust growth, and high product quality confidence, right? So fed batch. So fed batch is done in 2 steps. One is the 96 depot stage, and one is in the amber stage, which is an automated bioreactor. The culture conditions are maintained more in the automated bioreactor compared to your 96 depot, and then you basically evaluate your cells through the clones that you don't want, and then carry the ones that you want further along, and then come up with your top 6 cell lines. And then you have your product quality where you look at aggregation purity, various things like acidic peak, So you basically perform a whole lot of chromatography to look at all these different, you know, glycosylation, fucosylation, all these quality of your product. And then in-house, we also have like different sort of um, product quality validation. So for example, bioburden. So these cells needs to be free of any biological or any foreign animal particles, animal or non-animal, microorganisms particles. So basically for bioburden, you just put it on the agar and then read the CFU the next day and ensure that there is no bacteria growing in them. Then you have your mycoplasma checking using PCRs, and then you have your sequence validation using RNA sequencing, cDNA sequencing, and then you have all these other sort of sequencing as well, sort of testing as well. Right. So moving on to the last part of my talk, I just wanted to give you a brief overview of how the manufacturing looks like. You have your upstream, where you have your cell banks that I made earlier, right? And then you thaw that, and then you go on. So upstream is mostly cell culture. So you basically expand it from the inoculum stages up to the seed bioreactors and production bioreactors. Production bioreactors can be as big as 15,000 liters. It's a very maintained, controlled run where you have everything in place. All the dissolved oxygen, pH, everything is maintained. And then you basically start your harvest, and then it goes into downstream techniques, which is further more purifying the product antibody, basically using various chromatography steps where it passes through columns, filters, viral filtration, viral inactivation, ultrafiltration, diafiltration, until your final bulk drug substance, which is then sent over to the packaging site for packaging. Okay, and then in the last few minutes, I just want to give you a brief overview of like what are the different automation that is used in cell culture. For example, when I told you the theory for well plates expansion onto the shake class stages, you go through these high throughput liquid handling systems. It has the potential to do your plate transfers. It has the potential to do your like do automated cell culture, and it's very helpful because when you start with 8,000 clones, then it's very difficult to keep track of each clone as it moves along, right? So this makes it very easy for that. The major skills that you need for this is cell biology, a little bit of knowledge of drug development, some C programming skills, some SQL, and then a lot of Excel knowledge for that. For Amber 15, it's sort of like an advanced microscale bacteria. As you have seen this picture like multiple times today. It's a fully automated system. You inoculate and then you can leave. There's automatic addition of glucose, base, feeds, antifoam into the culture, and it reads all the metabolites, all the pH, all the cell count automatically. You can use this for parallel analysis or DOE analysis where you can have 48 different conditions and you can test them out all in a 15-day run. So it's very, very helpful. And for this, you don't even need a lot of knowledge. You just need some cell biology experience or knowledge, some knowledge about drug development, and some bioprocess or chemical engineering knowledge. And that's all that it takes. Lastly, there is process analytics also that is very helpful. So each batch that is developed is about $3 million. So it's very expensive process. So you have scientists and technicians watching this all the time. So process analytical systems allows you to monitor your runs so that if your run is going out of these like control limits, then you get a notification on your phone, on your email saying that something's wrong. And then when you double click on it, you can see that the glucose was overfed. So you can intervene immediately to fix your
fixture process. So for this, skills needed are some statistics, some multivariate data analysis techniques, linear regression, principal component analysis, those kind of studies, and then some manufacturing knowledge and cell knowledge. So all in all, that was my talk. Sorry, I'm on the top of the hour. I can see it. Sorry, I was kind of slow. So I can answer any questions that you have. I'm okay to stay longer if the participants are. So it looks like I have a few questions in the talk. Oh, Shyam.
Can you tell us how much time it will take more for you to explain the slides?
Sorry.
Can you tell us how much time it will take more for you?
Oh, no, I'm done. I'm done.
You're done.
That's it. Sorry. Yeah.
It's okay. Thank you, Mr. Bijay, for your very informative presentation. Now
Yeah.
we shall move to question and answer session.
Sure.
If you can speak directly to Mr. Bijay for your questions, or if you are not comfortable with, you can also message your questions. We have already two questions in chat box.
I was reading Shyam's question, but for some reason it's gone now. I don't know how to get it back. I'm not very good with Zoom, so sorry about that.
It's okay. Now we have two questions in your chat box. First is, what is the nucleotide size of the heavy and light chain sequence?
It depends. It could be, so your whole vector is about 10 kilodalton, 10 to 15 kilodalton. And your heavy chain light chain sequences could be, anywhere between 700 base pair to 1500 base pair.
Okay. And we have also got a second question. And the second question is, how much of the workflow in the cell line development process is automated?
Sorry, I didn't mean that. I meant base pairs. Sorry. Yeah.
Okay.
So what was this second question?
Sorry, Mr. Bijay, I could not understand what you said.
What was the second question that you had?
Okay, the second question is, how much of the workflow in the cell line development process is automated?
Quite a bit. So let me go back to the picture here. So all these yellow and green ones, I made these in a hurry, so I didn't put the tags here. But all these are automated. So basically, once you sort the cells, everything else is automated until you go to shake flash. Shake flash is very hard to automate because, you need some sort of system to, take the flash in and out of the incubator, which is hard to do. But then when you run the bioreactors, that is automated as well. So for the most part, it's very much automated, I would say.
Okay, I hope Sam, you got his answer. Do you have any other question from anyone? Okay, I have got one question for you. Like
Sure.
You said you choose most of those CHOS cells for your research. Is on what basis on what basis do you select the host cell? Is CHOS cell able to give result for most of your design projects?
The cell line is chosen very initially when they are doing research. They apply multiple cell lines. They grow these cells in multiple cell lines and see which one gave the best cell culture performance, growth profile, and the titer or the antibody concentration. So basically, that's what you're trying to see. So that's how they lock the cell line, pretty much.
Thank you, Mr. Bijay. I think we have no more questions. So we can end the section.
Okay.